And I also want to note that this presentation includes a very limited amount of the data directly related to the cancer, but I do believe that our research has a greater potential for collaboration with many of you guys. And our group has studied a small protein called Tribbles 3 or TRB3 in skeletal muscle and white adipose tissue. And today I'm going to share with you the story in skeletal muscle.
Maybe. Got a little bit of a lag here, I'm sorry. Nope. Didn't make this easy for me, did I?
Finally. So everybody here know that the importance of the skeletal muscle. And skeletal muscle is a highly malleable tissue, meaning that they adapt its structure and function depending upon internal or external stimuli. And one of the most important muscle adaptation is skeletal muscle mass, which is determined by the dynamic balance between protein synthesis and protein degradation. For example, IGF-1 or resistant exercise or anabolic steroid administration will increase protein synthesis over degradation, leading to increased protein muscle mass, muscle hypertrophy. On the other hand, aging, inactivity, or some pathological conditions will promote protein degradation, leading to decreased muscle mass, also known as muscle atrophy. Skeletal muscle is the single largest tissue in our body, consisting of about 40% of the muscle body mass, and the major tissue for the energy substrate and utilization and storage. For example, if you have improved muscle mass, then you would expect to have increased glucose uptake, insulin sensitivity, fatty acid oxidation, mitochondrial contents, etc. Whereas if you have impaired muscle functions, then you expect to have decrease in all of these ones. Therefore, physical activity is highly associated with preventing and or delaying progression of various chronic diseases, such as type 2 diabetes, obesity, and cancer. On the other hand, if you lose the muscle mass, this has been highly correlated with the motility and motility in various clinical conditions. For example, in this study, they look at the survival rate of the cancer patient. Cancer patient showing no cancer cachexia, mild cancer cachexia, or medium cancer cachexia, or very severe cancer cachexia. As you can see right here, that they show the very decrease in the mortality, and therefore maintenance of the skeletal muscle mass is very important to prevent or delaying the further complication of various diseases, as well as the quality of our life. Cancer cachexia is involuntary loss of the skeletal muscle mass in cancer patients, which is not fully reversed by just traditional nutrition the support. It's been known that about 50 to 80% of advanced cancer patients eventually get the cancer cachexia, and this particular cancer type include pancreatic and lower and the upper gastrointestinal cancers. Cancer cachexia is responsible for 22% of the death in cancer patients. So they do not die because of the cancer, but they die because of the loss of the muscle mass. Well, now we know that it's very important issues. However, there's a highly effective treatment do not exist. That's a really key important problems. The common feature of the cancer cachexia include the negative protein and energy balance. Let me just begin to talk about the protein mass regulation then. Protein mass is balanced between protein synthesis and protein degradation pathway. And AKT is known to be a master regulator of protein turnover through the regulation of protein synthesis, mTOR, and protein degradation, FOXO signaling pathways. Therefore, if you activate AKT, then you can have increase in protein synthesis, and also you can prevent muscle atrophy. However, what we do not know yet is what is the key regulator for this AKT, therefore regulating this protein turnover. We have studied a small protein called Tribbles homologous 3 or TRB3. This is a urokinase containing the conserved protein kinase domain without kinase activity. The way how TRB3 works is binding to the partner protein and regulating their functions or activity. This TRB3 is known to found in multiple tissues in response to stress, such as ER stress, starvation, or some pathological conditions. One of the best known function of the TRB3 is TRB3 binds to AKT and inhibit its phosphorylation, meaning that activity. In our previous study, we took the TRB3 knockout mice and the wild type animals, and then we put them on the high-fat diet. As you can see here, TRB3 knockout mice gained the less weight than the wild type animals. And when we challenged them with the glucose tolerance injections, that they showed the improved glucose tolerance, meaning that they have better glucose metabolism. We isolated muscles, and then we incubate them plus minus insulin, and then we saw that the TRB3 knockout mice showed better AKT phosphorylation in response to insulin, as well as FOXO signaling. Well, increased FOXO phosphorylation, meaning that inhibited FOXO activity. And we also overexpressed, so now going back, so we also overexpressed the TRB3 in the myotube, and we saw that IRS1 phosphorylation, as well as AKT phosphorylation was significantly impaired, meaning that the TRB3 inhibit AKT activity in muscle. In that study, we conclude that the TRB3 in the skeletal muscle inhibit AKT and therefore suppress glucose metabolism in mouse skeletal muscle. So before we getting into our recent study, I'd like to just remind you guys the AKT and protein turnover again. AKT, again, as I said earlier, regulates both protein synthesis and protein degradation pathway by regulating mTOR and the FOXO signal. If you delete the AKT, then you would have impaired muscle growth and impaired glucose metabolism. On the other hand, if you activate AKT, then you would have muscle hypertrophy. If you're looking at the multiple atrophic conditions, then you commonly seen that the AKT activity is downregulated. However, we don't know yet how the atrophic conditions can suppress the AKT activity. That's the one key question that remains to be answered. So our hypothesis is that the TRB3 will decrease protein synthesis and increase protein degradation by regulating AKT and its downstream protein. In order to test the idea, we first took out TRB3 transgenic mice, so overexpressing TRB only in the skeletal muscle, and then the mice showed no change in the body weight. But when we took out the 4 different muscles, then all the muscles we examined, TRB3 overexpression decreased the muscle mass. The decreased muscle mass was also associated with impaired muscle functions by assessing grip strength test and the running distance test. when we isolate the muscle and then measure the mTOR and then FOXO signaling, we saw that the mTOR phosphorylation was significantly decreased in the transgenic mice, and the S61K, a downstream for the mTOR, was also decreased in the transgenic mice. And we also examined the FOXO signaling, and the FOXO phosphorylation was decreased in the transgenic mice. And again, the FOXO phosphorylation, meaning that not many FOXO can get into the nucleus, and therefore they cannot activate the downstream. So one of the downstream for the FOXO is the atrogens, meaning that the proteins that are involved in the protein degradation, and such as atrogen 1 and MNRF1. These proteins are elevated in the transgenic mice, which are consistent with the data that I show you here that FOXO phosphorylation was decreased. However, the protein degradation protein that is independent of the FOXO signaling was not changed, meaning that this is mainly due to the regulation of the FOXO signal. So in this data, in this study, we found that the TRB3 decreased protein synthesis, but increased protein degradation. So you may wonder at this time that what if we delete the TRB3, then can we see the better muscle mass? To answer the questions, that we again took out the TRB3 knockout animals, and now we measure the muscle mass in tibialis anterior muscle and gastrocnemius muscles. As you can see that it's not huge, but there's a significant increase in the muscle mass. Just in case this is just one single muscle, you see that 10 to 20%. Guess what? You assume the whole muscle body 10 to 20%. That's a huge amount of increase. And this TRB knockout mice showed the greater increase of FOXO phosphorylation to isotypes, and then decreased atrogen 1 expression, meaning that the protein degradation pathway seems to be decreased in the TRB3 knockout animals. We do have a lot more data, but collectively we believe that if you're knocking out the TRB3, then you have increase in the protein synthesis, but decrease protein degradation, therefore you have more muscle mass. Well, this is all study we done in the normal and resting conditions. What if we actually challenge them with atrophy conditions? Can we see something greater change? So that's the next idea we thought. And the first atrophy condition we tested was simply fast mice for 48 hours. That's a very easy way we can induce muscle atrophy and see what happens. So first we took the TRB normal mice and we fasted them 48 hours. And as you can see here that multiple skeletal muscles were significantly decreased by just a simple 48 hours of the fasting. And interestingly, when we isolate the muscle from fasted mice and measure TRB3 expression, we saw that the skeletal muscle TRB3 was threefold higher than the wild type animals. That's very exciting. And then we also isolate and the muscles and then measure the AKT and the FOXO signaling. And as you can see that the FOXO AKT phosphorylation and the FOXO phosphorylation was all decreased, and atrogen 1 and MNRF1 was all elevated. This is exactly the same as what we did see from TRB3 transgenic mice. So we are very excited about that, and we move forward to look at that. And the next question we wanted to address was, okay, now fasting seems to increase TRB3, then what is the mechanism? So again, as I said at the beginning, TRB3 is regulated by the multiple stress conditions. So we first tested what if ER stress. So this is a very easy and simple experiment. So we isolated myotubes and we incubate them with the media or PBS. So this is a very traditional way that we can mimic the fasting in cultured cell lines. And we also incubate them plus minus with the full PBA, which is known as the ER stress inhibitor. And just to make a long story short, I don't want you to look at all this data, but from this experiment, we found that if we suppress the ER stress, then the fasting induced all the genes were just reversed to the normal conditions, meaning that all the fasting induced TRB3 expression or ER stress were TRB3 expression or the atrogen activation was all mediated by the ER stress. And now we just took out TRV3 transgenic mice and then we fasted them for 48 hours and we measured the atrophy, muscle atrophy, and both wild type and the transgenic mice showed the muscle atrophy. However, TRV3 transgenic mice showed even greater muscle atrophy, fasting-induced muscle atrophy than the wild type animals. And now we're back to looking at the TRV3 transgenic mice, knockout animals. And then they showed here, not really nice story, but at least the 2 of the 4, we saw that the TRV3 knockout animals were protected from fasting-induced muscle atrophy. From this result, we found that food deprivation decreased protein synthesis, but increased protein degradation. And we believe that fasting condition, this phenomenon is mediated by TRV3, which is increased by ER stress. So based on this result, now I'm going to show you just one piece of data that I believe to be very related to the cancer, cachexia. So in collaboration with Dr. James Carson at the UT Health Science Center, we actually isolated the quadriceps from APC mean mice. Some of you guys may know, but APC mean mice is not well-established colorectal cancer model, and it's also well-established cancer cachexia models. And we isolated quadril muscles from the wild type and the mean mice. And as you can see here, that TRV3 expression was significantly elevated by in the muscles from the APC mean mice. And I don't have the data here, but our preliminary data, we also found that if we implant LLC, Lewis lung cancer cells, under skin nearby the quadriceps muscle, and we induce the cancer cachexia. That's another way we can actually mimic the cancer cachexia model. And we isolate the muscle after that, and we saw that a similar result. But that's quite a preliminary, so I did not include the data here. So this data, even just a piece of data, we believe that this is a very promising data that TRV3 may be important for cancer-induced muscle atrophy. And with that idea, we also wanted to see that, okay, so then we saw that the TRV3 knockout mice is good to protect muscle atrophy. So how are we gonna actually inhibit TRV3 in humans then? We cannot generate the knockout humans. So how are we gonna suppress the TRV3 expression in human then? So one of the approach we did was dietary supplement. So here is a data we showed that the leucine supplement. So we actually give the mice with the leucine or the vehicles, and then we're looking at the TRV3 expression in the muscle. But we also generated 2 different atrophic conditions. One is the denervation. So it's kind of neuromuscular degeneration model. If you are the neurologist, then you disagree with me, but this is a very simple way we can generate the neurodegenerative muscle atrophy. And the other way is we simply immobilize the muscle, so then they cannot move, and inactivated conditions, so then they can lose the muscle mass as well. So both ways, we saw that TRV3 expression was robustly elevated, but this increase was completely abolished by the leucine supplement. This data clearly suggests that the leucine can effectively suppress the TRV3 expression in the muscle, and therefore we will be able to see the knocking out the TRV3 effect with this leucine supplement. So we will, based on this result, we are proposing that the cancer cachexia induce the TRV3 and leucine suppress the TRV3 expression. Therefore, they are regulating AKT, therefore protein synthesis and protein degradation pathways, and therefore we will be able to maintain the muscle mass better. And because of time, I'm just skimming the summary, but in conclusion, we believe that the fasting induced the TRV3 through the ER stress, which inhibit AKT and therefore suppress mTOR protein synthesis pathway, but activate the FOXO and estrogen expression and therefore protein degradation. Therefore, we get the muscle atrophy. Thank you for your attention. And this is the last slide, just citing our collaborators and the funding resources. Thank you for your attention.
Thank you, Dr. Ko. That was an excellent presentation. And also one of the very interesting topics in the field of cancer. Now, open to questions. When you're preparing for questions,